Hello friends, welcome back to my YouTube channel Nesting Padasale. So this video is about the shift 1 question. So I just made an important analysis of shift 1 question. So make yourself clear about the question as well as answer. So all the question I just collected here and there. So if there is any mistakes while typing questions or if I typed any question in a wrong way, please forgive me. Okay, and if you remember any question for shift 1 and shift 2, please type the question entering in which shift the question has been asked so that I can prepare the answer and I will be uploading in this particular video. So first question is from developmental biology. Which one of the following is correct regarding mammalian development? First is a zygote is a totipotent stem cells. Yes, it is a correct. Zygote means it's a fertilized egg. So zygote will lead to a formation of all type of cells. In addition to that, this zygote is also leading to the formation of extra embryonic cells. Okay, so zygote is the totipotent stem cells. Okay, and next, pluripotency of inner cell mass is due to OCT4, STAT3, NANOX. So this OCT4, STAT3, NANOX, CDX3 all are transcription factor. Okay, if you remember this one diagram which is given in Gilbert book page number 302, then it will be easy. So this diagram tell about the proposed function of NANOX, OCT4 and STAT3. Okay, and also this OCT4 will stimulate the morula cells which will be retaining it to become an inner cell mass and not trophoblast. Okay, and also NANOX will be working at a next differentiation event which will be preventing this inner cell mass cell from becoming hypoblast and promoting their becoming a pluripotent embryonic epiblast. Okay, and with regard to STAT3, the role of STAT3 is that it is involving self-renewal of this pluripotent stem cells. Okay, so second option is also correct, and CDX2 facilitates OCT4 and SOCT2. Okay, so here they had given what is the role of CDX2. So CDX2 is expressed in trophoblast and it will be preventing the OCT4 and NANOX expression. Okay, thereby stabilizing the trophoblast lineages. So the CDX prevents the role of OCT4 and SOCT2. Okay. Next question is regarding which technique is used to distinguish between alpha helix, 3'10 helix and pi, that is pi helix. Okay, so all this alpha helix, this T helix belong to a secondary structure of protein which is found in proteins as well as polypeptide. So if you understand this concept, then it is easy for you. So then arises the question, which technique is used to determine the secondary structure of protein? The answer is circular dichroism spectroscopy, that is near UV circular dichroism spectroscopy is used, whereas if you want to work on tertiary structure means you need to go for X-ray crystallography. Next, which organelle listed below is a non-membrane bounded organelle? So below I had given you explanation of membrane bounded organelle and non-membrane bounded organelle. Membrane bounded organelle mean they will be include a cell organelle which will be enclosed by a membrane, whereas non-membrane organelle they are not enclosed with the outer membrane. And this membrane bounded organelle they have a control of substance that they enter and leave the cells, and also this membrane is nothing but it's a phospholipid bilayer which are selectively permeable. Okay, and the inner structure of this membrane bounded organelle will be filled with fluid, whereas the inner structure of non-membrane organelle is filled with the solid. Okay, an example of the membrane bounded organelle include your nucleus, chloroplast, mitochondria, Golgi apparatus, peroxisome, lysosome. Example of non-membrane organelle include ribosome, centrioles, cytoskeleton and cell wall. And the answer is that peroxisome is example of membrane bounded, so it's not. Endosome, what is mean by endosome? Endosome is nothing but it's a heterologous collection of organelle that function as sorting of protein. It includes endosome include Golgi apparatus, lysosome and this vesicle. Okay, and Golgi apparatus, lysosome all are belong to this membrane bounded organelle. So endosome is also wrong option, and nucleus is again a membrane bounded organelle. And with regard to centrosome is a subcellular non-membrane bounded organelle. So centrosome, you can hear the centrosome while studying this microtubule organizes center. So correct option is centrosome. And which of the flower organ identity gene lead to flower with sepal, petal, sepal? That is, this plant doesn't have stamen. It don't have no stamen and no carpel. So you had studied about 3 types of gene. That's ABC model, type A gene, type 3 gene, type B gene and type C gene. Okay, only if there is any mutation in type 3 gene, then it will be affecting the stamen and carpel. And this plant, so here you can able to see this kind of plant which have a mutation in type 3 gene will not have stamen and carpel. In the places of stamen and carpel, there will be a petal. That is, the petal will be formed instead of stamen and sepal will be formed instead of carpel. And the example of type 3 gene is the agamus gene. So answer is agamus. Next, regarding sperm and egg activation, so you need to choose the incorrect statement. Okay, so capacitor sperm interact with the egg. Of course, it is the correct option. So capacitation is a process in which sperm will undergo a series of cellular changes that will be enabling the sperm to participate in a process called fertilization, and this capacitation occurs in uterus. Next, calcium play a role in egg activation. Of course, correct. Calcium play an important role in activation because this calcium will break a meiosis imposed developmental path. So you all know egg will be arrested in the metaphase stage of meiosis number 2. So this calcium ion play an important role in activating the egg. Okay, in addition to this calcium is also involved in activating our sperm, that is, it will be activating ATP generated apparatus and dynein so that it will be moving the sperm. Next, sperm can activate the egg without calcium. No, sperm without calcium, they are saying this is an incorrect option because sperm can activate a egg only in the presence of calcium. Like here you can be able to see it's a given developmental biology, the sequence is given, and here the role of calcium in the sea urchin sperm it had given. So here you can be able to see the influx of calcium ion to calcium ion channel will activate the sperm motility, and then sperm will swim up to the result gradient towards the egg. Okay, so without calcium sperm cannot able to move towards egg. Next, egg secretes some attractant for sperm. That is absolutely correct. So here you can be able to see first there is a hemoattraction of the sperm to the egg by soluble molecule which are secreted by egg. So egg will be secreting a soluble molecule and that will be creating a hemoattraction which will be attracting sperm. Okay, so these things you should know. Next thing is with regard to critically endangered animal which is found in eastern parts of Russia that normally live alone and might live with a mate and help in offspring upbringing. So the answer is Amur leopard. So Amur leopard is a critically endangered organism which has given critically endangered by IUCN Red List on 2007. Okay. And next, which type of forest cover India's largest area? The answer is tropical deciduous forest. Both tropical moist forest followed by tropical deciduous forest. Nearly 60% of area will be covered by the tropical deciduous forest. Then there is a question with regard to gibberellic acid signaling pathway, and they are asking we choose the correct statement. Okay, so here you can be able to see follow this particular article, friends. So in this part they had given clearly about it. So gibberellin. So here you can be able to see the gibberellic acid receptor is gibberellin insensitive 1. So following a gibberellin hormone binding, the soluble GID1 protein will be interacting with the DELA. DELA is a growth repressor. So in the absence of GA, the DELA protein will be repressing the seed germination and lowering the plant growth. Okay, now all the gibberellin dependent pathway, whereas in the presence of gibberellin, what this GID1 will do means it will be interacting with the DELA protein and then it will be leading to proteasomal degradation process. So what is the DELA protein helping GID1 degradation? That's a correct option. Next thing is that GA is a negatively regulated signaling pathway. Of course, gibberellic acid pathway is a negatively regulated pathway. And next is GID1 join with DELA helping degradation. So here you can be able to see this GID1 once the gibberellic acid has bind to this receptor, then it will be binding with DELA, activating them to lead to proteasomal degradation. Okay. Next, geological time scale. So what is the correct order of geological time scale? So geological time scale is a calendar for even in Earth's history. It is subdivided all time into named unit of abstracted time called in descending order of duration, saying that first aeon, followed by eras, followed by periods, followed by epochs. So correct option is that aeon, era, periods, epoch. Okay. So next, species richness depend upon. So I think this particular thing, the option is wrong or not, I don't know. But only if a correct question paper has been released, only then we can able to know all those things. In one thing they had said species richness depend upon only on number of species and it does not depend upon abundance of species. But in another website, I could able to see species richness depend upon both the number of species as well as abundance of individual of those species in a particular community. So option number A and option number C are correct. Next, in an enzyme, the PFK, the site where ATP bind is mutated, then what is the effect of PFK? So PFK means what? It's a kinase enzyme, that is phosphofructose kinase, that will be phosphorylating the fructose 6 phosphate to form fructose 1,6 bisphosphate, and this PFK is an important regulatory enzyme in glycolysis. So you should know this PFK is allosterically inhibited by ATP and it is allosterically activated by ATP. So ATP is a natural allosteric inhibitor of PFK. So generally generally it is acting as inhibitor in order to prevent the unnecessary production of ATP through glycolysis. So they are saying when the site of the ATP binding is mutated means then ultimately ATP cannot be able to regulate the PFK activity. As a result, the PFK cannot be inhibited, and what it will be there. So please forgive me for this mistakes. I didn't correct my PPT, so that's why. So sorry friends. So what they are saying, PFK is not inhibited, then ultimately the activity of PFK will start to increase. Next, India has which type of ecosystem? So India has alpine grassland, which is otherwise called as tundra ecosystem. Okay, so India has generally consists of 3 biodiverse hotspot, friends. That is, it consists of Western Ghats, Himalayas, and Indo-Burman region. Okay, this taiga, another ecosystem, which is also called as boreal forest, is generally present in America, Europe, and Asia. Okay, so the correct answer is alpine grassland or tundra. So still I am not sure about this particular thing. If you have any other options, please tell in the comment section, so that many students will be benefited. Observed monosaccharide by intestine. So from intestine or from stomach, how does this monosaccharide will be observed and excreted to? Okay, so the correct option, many students are collapsing between GLUT4 and GLUT2. The GLUT4 is present not in this particular process. GLUT4 is present in adipose tissue, skeletal muscle, brain, and heart, whereas this GLUT2 is responsible for transporting dietary sugars like glucose, fructose, and galactose. So there is an article published in 2005 in Journal of Physiological Biochemistry, and here they had given role of GLUT2 in dietary sugar handling. So GLUT2 is the correct option. So if you want more in-depth analysis, you can refer this particular video. Next, endocrinology, pituitary gland. So they had given pituitary gland and their location. So following which is called as incorrect combination. So this particular thing already we had discussed while our vision series, friends. So in that you can able to see gland and what are the hormones that are produced by that gland and the major function. So these things you can refer, and if you know the answer, if you know the question, then please type the full question if you understand the question, friends. Okay, so this oxytocin and vasopressin are secreted by posterior pituitary and prolactin is secreted by anterior pituitary. Okay, so here I had given this kind of thing. And next, I think one question was been asked from hunger hormones. I already, when I was doing this revision series, I just mentioned you ghrelin is a hunger hormone, and this particular hormone regulates the glucose homeostasis by inhibiting the insulin secretion. In addition to that, this particular hormone also regulates gluconeogenesis and glycogenesis, and this hormone decreases thermogenesis. Okay, next, a scientist was investigated 2 young birds. One was born with down feathers, open eyes, and they are free to fly. Other bird has a closed eyes and they were dependent on parent and named them. Okay, so there are 2 type of thing, altrical and precochial. So parrot, pelican, songbirds are belong to altrical, and the chick, duck, rhea belong to precochial. Okay, so what does mean by precochial bird? Mean it is a capable of moving around on its own soon after the hatching. It is derived from a Latin word called precochius. Okay, and altrical means it is incapable of moving around on its own soon after hatching, and it comes from a Latin word called as nourishing, with a reference that these kind of birds need extensive parental care. Okay, so here they had given one bird was born and they are free to fly, which means the bird is precochial, and next other bird has a closed eyes and they are dependent on parent. They need a parental care, so that bird is called as atrical. So you can refer these slides for your future references. Next thing is that match the following with their thing. Okay, so this is with respect to cell cycle checkpoint. They had asked this particular question, friends. So they are asking you to match the following. So first one is this CDK1. So CDK1 play an important role during G2 to M phase transition, friends, and CDK2 had a role in G1 to S phase transition. In addition to that, CDK2 is also involved in regulation of S phase, and it is involved in initiation of DNA synthesis. Okay, and with regard to Aurora B is involved in the process of mitosis, and it is involved in chromosomal segregation, and this Aurora B has been formed before early to mitosis, and it is an enzyme which is also called as mitotic checkpoint kinase. And APC, that is anaphase promoting complex, it is an E3 ubiquitin ligase enzyme, and it will make a cell cycle protein for degradation by 26S proteasome complex, and this anaphase is important for transition from metaphase to anaphase. In addition to that, it also promotes the exit of cell from mitosis and cytokinesis. So this APC is mainly involved in cytokinesis process. Next, the drug antimycin inhibit which component of electron transport chain? So the drug is synthesized as a secondary metabolite by Streptomyces bacteria. It inhibits the flow of electron through complex number 3 of electron transport chain by blocking the passage of electron from site B to site C. Okay. So the next question is with regard to ecology. So which type of species are invasive in climatic community? What is mean by invasive? Invasive species are non-native species, and what is mean by climatic community? Climatic community is filled with climatic species, and the other name of climatic species include late cirrus, rate succiona, K-selector species belong to this particular climax community. That is, the plant which are having present in this community will grow with a limited resources. Okay, so the correct answer is K-selector species. So if you refer the R and K selection video which I had uploaded under ecology unit notes, here you can be able to see. So climatic communities are stable communities. So generally in a stable community, K selector species will be highly present. So generally K selector species are became invasive in the climax community. Which next thing is organic compound nitrogen converted by bacteria in which form? So bacteria will convert a nitrogen that is getting present in a soil to ammonium ion, and this ammonium ion will be taken by the plant for their thing. Okay, so organic compound nitrogen converted bacteria is NH4 plus. Okay, and next thing is the nitrification and all, converting ammonium ion into nitrate ion, and denitrification means converting nitrate ion into nitrogen gas. Next, ammonification means again this ammonification process is also carried by bacteria, where the bacteria will be breaking down the nitrogen containing compound which is present in the dead organism into simpling substance like ammonia. Next, which of the following is not a tyrosine derivative? So if you check on this particular same revision notes for unit number 7, system physiology animal, here I had given a catecholamine, that is neurotransmitter biosynthesis. So from tyrosine, dopa is manufactured, and dopamine will be manufactured from dopa, and norepinephrine is produced from dopamine, and epinephrine is produced from norepinephrine. So from tyrosine, dopa, dopamine, norepinephrine, epinephrine are produced. Okay. So the correct option here is serotonin, which is not derived from tyrosine. Okay. Next is they are given a precursor and they are asking a final product. So first is pyruvate. So when a muscle degrades amino acid for the energy need, the resulting nitrogen is transaminated to pyruvate in order to form alanine. So this process is being performed by an enzyme called alanine transaminase, which will be converting the L-glutamate into alpha-glutarate and pyruvate into alanine. Okay. Next, oxaloacetate. So oxaloacetate produced in, that is, if you study this malate aspartate shuffle process, you can be able to answer this question. The oxaloacetate produced in mitochondrial matrix can be converted into aspartate by again transamination reaction catalyzed by aspartate aminotransferase, which is coded by a gene called GO2 gene. Okay, and next is hexose phosphate. Hexose phosphate I couldn't able to get any article, but in this article which has been published in 1957, they had mentioned the role of hexose phosphate in the synthesis of cellulose by Acidobacter xylenum. If you want more in-depth analysis, you can go. And pentose phosphate pathway acts as a precursor for formation of nucleotide. So if you remember the pentose pathway, here you can able to see it will be giving rise to ribose 5-phosphate, which is a basic structural unit of nucleotide forming in RNA and DNA. Okay, which of the following is true for plasmodesmata and plasmodesmata transport across plant cell? So you should know about two important pathways, symplast pathway and apoplast pathway. Symplast pathway is a pathway in which a water molecule will be moving through the plasmodesmata region into the cell, whereas an apoplastic pathway is a path in which a water is moving between an intracellular space. And the options are plasmodesmata is present only in symplast and cell wall is present in apoplast. The answer is absolutely correct. Apoplast transport occurs through plasmodesmata. No, apoplast transport occurs through this kind of intracellular space. Okay, next symplast pathway occurs through plasmodesmata. It is also a correct option. And next, transmembrane transport occurs through tonoplast. No, it is a wrong option because tonoplast is generally involved in maintaining a turgor pressure of plant. Next, match the major crop with the country of origin. Okay, so banana is native to Africa, and mung bean is native to East Asia, and wheat is native to India, but I'm not sure about this particular thing. Wheat is native. Some are saying that wheat is native to southwestern parts of Asia, and Aryan brings the wheat to India, like that history is saying. I don't know. If you know the correct answer, tell me in the comment section. And sorghum again I say sorghum is native to Africa, like that many are saying. I don't know. And also why they had given Indonesia, I don't know. Only if correct question arises, we can able to discuss. Next things with respect to match the following. This has been asked from ecology. So first is that aposematism. So it is a advertising signal of an animal to a predator that is neither worth for attacking and not eating. So this is include toxicity of an animal, venom of animal, and smell of animal, sometimes bright color and distinguishing wing pattern. So the correct example is absolutely coral snake is a correct option, because the coral snake will be having its phenomena and it is black band and orange gray color will be there. So this is a warning signal. Next, mimicry. The example is monarch butterfly. butterfly. Monarch butterfly is the best example of mimicry. Both the monarch butterfly and viceroy butterfly will be co-mimic each other. One is palatable to eat and another one is not palatable to eat. So birds will not able to take this particular butterfly, so they can evolve side by side. And bioluminescence, so nearly half of the jellyfish will be producing this bioluminescence. So bioluminescent example is bright jellyfish. Next is aestivation. Aestivation means dormancy of an organism during a hot and dry environment. It is opposite to hibernation, which it is due to dormancy of animal during winter. And the example is African lungfish. So African lungfish will undergo aestivation during summer season. So they will be going in a deep into the sea. They will be forming a mud cocoon where there will be no food, no water. For 5 years period it will be following a aestivation. And which of the organism having a types of cleavage? If you remember this one diagram given in the Gilbert book, then you can easily answer this question. First, isoleucithal egg, they have a spare and evenly distributed thing. So isoleucithal egg will follow a radial cleavage. Example is endoderms and amphibian. And spiral cleavage example is Annelida, molluscan platform. And bilateral cleavage example is tunicate. And rotational cleavage example is mammals and nematode. And mesoleucithal egg, moderate vegetative yolk dispersion, and the cleavage pattern is dispersed radial cleavage. Example is amphibian. And meroblastic incomplete cleavage, two types of eggs are there, teloleucithal and centroleucithal. Teloleucithal means dense yolk throughout the side of cell. An example of cleavage that is undergoing by these particular cells include bilateral cleavage. Example is cephalopod mollusk, and discoidal cleavage example is fish, reptile and birds. Okay. And centroleucithal egg, where the yolk is present in center of egg, and the cleavage pattern is superficial. Example is insect. So if you know these things, then you can easily answer this particular question. In which phase of cell cycle chromosomes are clearly visible? Chromosomes are clearly visible during the phase called M phase, which is also called as mitosis phase, and also in the M phase during the metaphase stage the chromosomes are highly visible. Okay, so generally for karyotyping studies and all, they generally take a metaphase chromosome, because in metaphase the chromosomal will be really in a condensed pattern, and during metaphase stage the nuclei will be disappearing and all chromosome will be shorter and thicker. They will be aligned in a equatorial pattern. So that's the reason why for karyotyping and all metaphase phase is taken. Which of the following does not apply to biological species concept? So biological species concept is generally applicable only to sexual organisms. It is not applicable to fossil animals, as well as it is not applicable to asexual organism. So cyanobacteria is the only asexual organism which will be reproduced by binary fission and multiple fission. So the correct option is that biological species concept is not applicable to this particular cyanobacteria. And this is with respect to few questions. I am currently working for some other question. If you have some other question in your mind with respect to shift 1 or shift 2, then please do tell me in the comment section. Type the question so that I can produce an answers. So part 2 of this particular shift 1 question is currently on way. So subscribe to my YouTube channel. So once I had completed solving those questions, definitely I'll update you. Thank you friends. Thanks for watching this video.